Good afternoon, everyone. As she said, my name is Meaghan Rose. I have had the pleasure of being in Eureka for 3 semesters now, and I work alongside my mentors, Dr. Meaghan and Dr. Masuoka, to identify and isolate bacterial endophytes from seeds with high and low melatonin content. So what is an endophyte? An endophyte is a microscopic organism, typically bacterial or fungal, that lives within a plant. These organisms are typically non-pathogenic in that they do not harm the host plant in any way. And in fact, in most cases, these organisms actually benefit the host plant. And in some cases, they can produce antibiotic compounds that will help the plant resist potential pathogens. And they can also help in the acquisition of nutrients and water acquisition for the host plant. The plant, in turn, provides a controlled, stable environment for the endophyte to flourish in, and also provides energetic compounds such as glucose for the bacterial endophytes. Now, the benefit of actually having these endophytes is very vast, because each of these endophytes provides a different characteristic. Each of them can provide nutrients, water, antibiotics, and so if you have a higher diversity of these endophytes, then therefore the plant will probably have a higher survivability rate. Melatonin is a well-known hormone that induces sleep and reproductive cycles in animals. But in 1995, it was discovered that plants also produce melatonin. Now this is a little unusual because plants don't sleep, so why would they need it, right? So they looked at it and they actually found out that melatonin induces cellular growth and tissue repair in plants, which is very beneficial. But melatonin is also in higher concentrations within the seeds. This is a little unusual, so we sort of wanted to see why it was so high. It's such a high amount that it seems a little bit unlikely that the plant was able to produce this hormone alone. Perhaps during germination. During germination, there is a bit of fermentation in which, because the seed is actually enclosed from the environment, therefore there's no oxygen. So in order for the embryo to actually start growing, it has to metabolize under anoxic conditions without oxygen. But the problem is whenever you're doing this process, you actually produce compounds that can be toxic, and they can actually damage the embryonic cells. But melatonin, perhaps, has shown characteristics to be antioxidant, so maybe during this germinative fermentation, melatonin is actually acting as a buffer to help protect the plant cells. The seeds that we analyzed were, we had celery, sweet corn, wolfberry, and white mustard, each of them having varying levels of melatonin. For our low melatonin groups, we did have the celery and sweet corn, with their melatonin contents being only 7 and 2 nanograms, versus the high melatonin who had 103 and 108. That is a big difference in terms of melatonin content, and the exact reason for this is not known. So as I said, perhaps these endophytes are helping to either produce the melatonin, or perhaps they are helping to produce precursors for this hormone. In order to isolate and identify the bacteria from the seeds, we had to ensure that all of our working conditions were completely sterile and aseptic so as to avoid contamination. In order to do this, we surface sterilized each of our seeds, and so since we surface sterilized them, the assumption is that anything that grows after that point came from within the seed, not from the outer environment. We crushed the seeds and grew them in a nutrient auger, BHI broth, nutrient medium, BHI broth, which is brain heart infusion, and it's just a common medium that is used for bacterial cultivation. Afterwards, after we were able to isolate our DNA, or isolate our endophytes, and separate them out based on color and colony morphology, we then analyzed their metabolic processes. And this is a way to differentiate between the different organisms and see if we can speciate them. To double check our metabolic results for identification, we then isolated the genomic DNA from the bacteria and took those sequences, information, and used that to fully identify the endophytes. For our metabolic results, after our metabolic testing, we found that none of them were able to degrade gelatin, but most of them were in fact gram-positive. With bacteria, you have two different types of bacteria. You have gram-positive and gram-negative. Gram-positive have a thicker cell wall, which enables them to survive in environmental conditions better than the gram-negative bacteria. So in most of our seeds, we had a higher rate of gram-positive bacteria, which was to be expected. Only a few of them were able to ferment lactose, which is just another form of sugar that they're able to take care of. But we did notice that for the celery, we had a total of 11 isolates. But looking at them and looking at the metabolic results, we realized that a few of them may actually be replicates of each other. So our real number for celery came out to 4 based off of the metabolic results. And for our high melatonin content seeds, we had totals of 9 and 8 for the wolfberry and mustard. Once again, none of them were able to degrade gelatin, and once again, most of them were gram-positive. In terms of the endophytic diversity, we found that the low melatonin content seeds actually had a lower level of diversity versus the high melatonin content seeds, which is sort of what we expected. Even though we tested 3 different types of low melatonin content seeds versus the 2 high melatonin content seeds, we still got a higher number versus the low melatonin content seed. In order to test to see if our diversity was true, our diversity count, we needed to fully identify each of these endophytes. So to do that, we had to pull out the genomic DNA. Bacteria have 2 different types of DNA. They have these smaller circular DNA called plasmids, and then they also have the larger chromosomal. They have the larger DNA that is referred to as the genomic chromosome. The plasmids are highly variable. They can change. Bacteria like to change them throughout. And so that way they can gain different abilities, such as antibiotic resistance, the ability to break down certain compounds, and stuff like that. But those things are highly variable, versus the genomic material, which tends to stay constant throughout evolution. And so that is how you were actually able to identify them, by comparing regions on the chromosomal DNA. So what we did is we used the Meaghan genomic DNA isolation kit, which is a common isolation kit that you can use. And after we isolated our DNA, we had to ensure that the DNA was fully intact, so that way we could continue on with our identification. And in order to do that, we ran an agarose gel through the electrophoresis apparatus that uses electrical charges, electrical currents, to separate the DNA based on its size. So here we have our agarose gel. On the left side, we have markers, which are DNA of known lengths that you can use as a reference point when you're referring to your unknown lengths. So here we have our 4 isolated DNAs. The bands are solid, fully intact, there's no smears, which indicates that the DNA in fact was intact and we had successfully isolated it. Now that we knew our DNA was fully isolated, we had to move on to the next step, which was the amplification of a certain gene. We wanted to use the polymerase chain reaction to amplify a common gene that is found within most bacteria, but this gene can have slight variations in it, which are specific to each species. And looking for those differences is how you are able to identify them. So we had to use a forward primer and a reverse primer, because what we were doing is we are not replicating the entire chromosomal DNA. Instead, we are only replicating a small section. So we have one forward primer that will replicate in one direction, and then a reverse primer that will replicate in the opposite direction. And doing it this way, you can double check yourself to make sure that your results are conclusive. After our PCR, we were looking for, once again, a smaller chunk of DNA, but we also wanted it to be amplified. We wanted to replicate it. So, in seeing this brighter band, the higher the concentration of DNA, the brighter the band will be. So yes, we did in fact fully amplify the DNA. It was brighter, therefore there was more. And the DNA actually moved to a length of 1.5 thousand base pairs, which is our expected length for that particular gene that we sequenced. Once we knew that our PCR was successful, we were able to send these sequences off to a third party, where they would actually sequence this DNA for us and send us back each individual base pair on this DNA. And so we would take that information and we would put it into the NCBI BLAST database, which contains the genomic material for all known organisms. So we looked at 4 of the celery isolates, and we found that 3 of the 4 isolates were in fact identical, just as we suspected. They were identical, even down to the strain. We had Erwinia girondensis, strain EM595. This was the common one. And then we also had Pentoia agglomerans, and we were able to identify that one to the strain as well, RYC17. So whenever I'm talking about the DNA, the genomic sequence that we analyzed, this is it. We analyzed the 16S ribosomal RNA gene, which is right here. As I was saying, it is highly conserved throughout all bacterial organisms, and so that's why it's such a good gene to use for identification purposes. The endophyte that we found, Erwinia girondensis, this strain was the strain that we actually identified, was only discovered this year in 2016, which was really interesting. So there's not too much information about this particular strain, but this species has been identified in over 3 countries. So this organism is capable of surviving in different climates and environments. It was isolated from palm fruit trees, but we isolated it from celery seeds. And this endophyte is capable of providing and solubilizing potassium, which can then be used for metabolic processes within the host plant later on. And this organism also has a high GC content within its DNA, which is an indicator that its DNA is very stable. So it is able to withstand high temperatures or perhaps even some levels of radiation. And something interesting though about this particular strain, the Erwinia species are typically pale white, but our species, our strain, was yellow. So this uncharacteristic coloration has been demonstrated to actually originate from another bacterial species called Pentoia. And so as I was saying before, how the bacteria can exchange their plasmids, I believe that is what happened here as well, and that the isolate that it was sharing the seed with, Pentoia agglomerase, actually shared the plasmid for it. Pentoia agglomerase is our other endophyte that we found. This is an opportunistic human pathogen, but it has also been identified and isolated in nearly everything. It's been found in water, soil, plants, animals, it's everywhere. This organism has been shown to be slightly tumorigenic in some organisms, but not all of them. So that exact relationship, we're not quite sure if it is actually harming the host plant, but it does aid in hormone production, we believe. So these organisms are able to produce glycol and catechol, which are precursors for pesticides and flavors and fragrances. And have you ever smelled celery? Celery has a very strong smell, so perhaps these endophytes are helping to produce that strong fragrance by providing these precursors. In the end, we did find that our high melatonin content seeds did have a higher level of diversity, which is what we expected it to be. In terms of agriculture, these endophytes, if we can fully understand their roles within the host plant, then we can actually use these endophytes as a form of a natural fertilizer for our crops. This has been demonstrated before with other species. Tomato plants have been spray inoculated, they sprayed the bacteria on them, and these tomato plants immediately showed root and shoot growth, as well as an increase in yield, production yield. So they had more tomatoes produced. And so if we can use these endophytes, if we can understand them better, then we'll be able to move away from these toxic fertilizers that are running off and polluting our water tables. Then if we could use these endophytes as a natural form, give the plants a way to fight the resistance off, or fight the pathogens off by themselves, find ways to survive in different environments. If you can take an endophyte that aids in water acquisition or aids in saline resistance, then you can take that same plant and plant it somewhere else that it would not have survived before. And so the same idea can be applied to many different crops, and the idea of doing this is still pretty new, so I think it would be really worth the trouble to look into it and see what they do. Because each of these endophytes is very diverse and they can do something amazing, so I think it's important that we look at them. As I said, we've only done 4 of our isolates. We've only identified 4, but we got 2 strains, or 2 identities. So we need to go back and identify all the rest of the endophytes from the rest of the seeds and see what their potential roles are within the host plant. We'll be doing the same method. We'll be isolating their DNA and using the polymerase chain reaction to amplify their DNA, send it off for sequencing, and use that to identify them. Thank you. Are there any questions? Yes, sir.
Meaghan, you said that one of your species is just recently discovered.
Yes, sir.
So, do you know anything about why it took so long to discover it, or why it's a new discovery?
Right. Okay. So this particular species, or this particular genus, Erwinia, has had a few new additions to its species count. This was only just recently, whenever they were doing more DNA analysis, and they found that there are actually new species that can be classified under here. So these new classifications are still pretty new. The difficulty in that is simply because the bacteria have a tendency to change. They have a very high mutation rate, and so that's why you end up getting all these different strains. So our particular species had been identified, but the strain was only identified this year. And so that's the thing, is with some bacteria you can have different strains which are capable of different things. So they might be the same species, but they can do different things. And so that's why you kind of have to take your time and really look at them. So it's pretty difficult to do the strain exactly. Yes, ma'am?
Because you said, human melatonin was identified with sleep.
Yes.
But do you think that the finding of melatonin in plants may have implications that maybe melatonin is doing more in humans than what we originally thought?
Yes, I believe so. I definitely believe so. And the fact that it is able to induce cellular growth and tissue repair, I think that it may be doing the exact same thing to us. Because whenever humans sleep, that is actually when our body is taking the time to repair things that have been damaged throughout the day. So perhaps by inducing the sleep, it gives it a chance to actually go in there and help fix whatever's been damaged, give the body time to repair and stuff like that. Yeah, that's right. That's right. Are there any other questions? Yes, ma'am.
You said that you have, for the future, you have to isolate, identify what you have. How many isolates do you have to identify?
In total, we had a total of 32 isolates, but that number has been reduced simply because we were able to group some of the celery together. So I would say that we probably have like 28 more left to go, somewhere around there. But the remainder of them are gram-positive. We were able to do most of our gram-negatives this round, so yeah. It'll take some time, but we can do it. Now that we have our methodology down, so. Any other questions? All right, I guess ma'am.
Just curious, if you have to name any, what would you say are the limitations of your study?
Are the limitations of my study?
Mm-hmm.
I would say that I can't really grow the plants and see how the endophytes are truly interacting with them. That's something I would really like to do, see if I can grow the plants and map the movement of the endophytes. So for example, inoculate the soil with a bacteria and see if that same bacteria ends up in the seed. Track that movement. I would really like to do that. And also in terms of like the exact role of melatonin, we can't really look at that simply because we don't have the equipment here, and I definitely don't have the expertise. But there's a few things, but I believe we're doing well with what we have for now. Thank you. All right. Anything else?
Let's thank Meaghan.
Thank you.